Record. Okay. So far in this course, we've been talking a lot about eukaryotic organisms, processes that take place, cell division, many different features and characteristics that are specific to eukaryotic organisms. So we're going to take the next few moments today to just take a look at some characteristics and features of both prokaryotic organisms and viruses. And so the things that you may have seen already at this point is that there are some unique features about these organisms as it relates to eukaryotic organisms, and especially the virus. Okay, that's one of the more sort of debatable. You know, there's a lot of debate on whether or not viruses are living things or not, or are organisms or not. And we'll take a look at why that is so today, as we know that viruses cannot carry out life's processes by itself, but it requires a host cell. So that leaves the question of whether or not they're actually organisms or living things. Prokaryotic organisms, we talked about them off and on throughout the semester when we compared cell types and so forth, but we're going to take a little bit more of a deeper dive at some of the characteristics that are unique to prokaryotes. And usually when we're talking about prokaryotes, we are referring to bacteria. All right, so we'll look at some features, and I'm trying to share my screen is what I'm trying to do. Give me 1 second. PowerPoint. We'll start with prokaryotes. I'm putting it up now. Share screen. There we go. All right, so you should be able to see my screen today. I want to highlight some very important points about prokaryotes in terms of their cellular structure and function. All right, so again, we've talked a lot at this point just about the two basic types of cells in terms of points of comparison. You should definitely be able to readily identify a prokaryotic cell versus a eukaryotic cell if I were to show you some type of image or a schematic. All right, there are things that should stick out to you right away in terms of being able to identify one or the other. All right, we know that the prokaryotic cell is a very simple cell. It's a lot less complex than the eukaryotic cell. Okay, over here, we know that the eukaryotic cell possesses a number of different structures and features that we do not find in the prokaryotic cell. So the nucleus, the endoplasmic reticulum, all of these membrane-bound structures or organelles that originated from membranes are going to be present in a eukaryotic cell, but we won't find those in a prokaryotic cell. Okay, so I want to highlight a couple of things in terms of the structure of a bacteria cell here. One is the presence of this cell wall and capsule. Okay, so if we look here where I have my cursor, this green part represents the plasma membrane. All right, now we know that the plasma membrane is a barrier that we know is common to all cells. Okay, all cell types are going to have this plasma membrane layer that separates the internal compartments from the external environment. Okay, so we have the plasma membrane here in green, but right outside of the plasma membrane is another layer or covering that we find on bacteria cells and some other organisms that provides another layer of protection. That's the cell wall. Okay, this cell wall is a very important structure in bacteria, as we'll see in just a moment. We'll talk a little bit about the peptidoglycan cell wall and how it's important in terms of identifying bacteria, you know, as either gram-negative or gram-positive, but that's another layer of protection. And then we have this outer covering right here that we call a capsule. This red capsule is also something that can distinguish prokaryotic cells from eukaryotic cells. All right, we see this tail-like structure right here. This is the flagellum. All right, this structure right here is necessary for a locomotion or movement in bacteria cells. All right, there are some eukaryotic cells that have them as well, so it's not necessarily unique to bacteria cells, but just in terms of overall complexity and structure, we can look at this bacteria cell and see that it's a lot less complex. It's rather simple. We talked about cell division in eukaryotic cells, and here we have binary fission. prokaryotic cells or bacteria replicate by means of binary fission. And we talked about this a little bit this semester. Binary fission is a form of asexual reproduction where the organism or parent cell duplicates its genetic material before segregating and dividing off into two daughter cells. Okay, so this is a form of asexual reproduction that only requires one parent. All right. Some other structures that are present in the prokaryotic cell are going to be plasmids and then this nucleoid region. So we know, we hear the term nucleus quite often in reference to eukaryotic cells, right? We know that that's where our genetic material is stored, in the nucleus. Well, prokaryotic cells don't have that structure, but we have what we'll call the nucleoid region. Okay, so there's a region within the bacteria cell that contains that circular genome, the DNA. Okay, and this nucleoid region is this open area inside of the cytoplasm, right? So if we're looking at this image right here, the blue area is our cytoplasm, which is basically the inside of the cell, right? And so we have this nucleoid region that contains our chromosome. Now, that's another point of distinction here. Typically, bacteria are going to have one chromosome, right? One circular chromosome. This is a defining feature that you should have in your notes here that distinguish it from eukaryotic cells that are going to be more complex, and it's typically going to be more genetic material, more DNA, more chromosomes present. All right, bacteria only have that single circular chromosome, right, as opposed to eukaryotic cells that are going to have linear chromosomes. And then, but bacteria also have plasmids. Plasmids are also DNA. They're circular DNA that are not necessarily a part of the genome for this bacteria. So we call it extra chromosomal DNA. And what these pieces of DNA do is they basically confer some types of advantages for the organism. Okay, advantages such as being antibiotic resistant, other features that basically enhances their chances of survival. Okay, they can range anywhere from 5 to 10 genes, right? So remember we talked about the genes, which are organized units within our DNA that code for proteins. These plasmids can also contain genes, and then these genes are going to be responsible for those properties that I mentioned earlier, such as antibiotic resistance. They can contribute to virulence in the host and so forth. Okay, so these plasmids are genetic structures that are found in our prokaryotic cells. We're clear about the cytoplasm here. We know that all cells are going to have the cytoplasm, and that's where we're going to find all of our internal structures, kind of like that gel-like matrix inside of the cell, and that's where most of the metabolic activity taking place, and growth, metabolism, replication. All of those things are going to take place inside of the cytoplasm, particularly on those ribosomes, right? So we've talked about ribosomes before, okay? Prokaryotes do have ribosomes. And we know that ribosomes are necessary for protein synthesis. That is going to remain true in prokaryotes. Okay. Some other points of comparison here. This plasma membrane, I touched on it a few moments ago, but I wanted to highlight again this plasma membrane. All right. We know the most important role of this plasma membrane is to separate the cell, the internal environment, from the external environment. All right, and we spent a great deal of time talking about the structure of the plasma membrane for any cell. We said that this structure is we refer to it as a fluid, a phospholipid bilayer. Okay, so this plasma membrane is made up of two layers of phospholipids interacting together to kind of form that bilayer or that sheet. All right. We talked about that structure, or one characteristic of the plasma membrane being amphipathic. Okay, this amphipathic phospholipids have that hydrophilic head and the hydrophobic tails. And so we had the arrangement of that lipid bilayer where the heads are, facing outward, interacting with the aqueous environment, but the tails are buried on the inside. So you'll recall that as you start to study the prokaryotes. All right. We also know that inside of the plasma membrane, there are proteins. All right. So we said that the plasma membrane of cells are mostly made up of lipids and proteins. This doesn't change here. Another point I wanted to highlight here was the fact that this membrane is semi-permeable. We talked about that concept. Okay. What does semi-permeable mean? Anyone? What does semi-permeable mean?
Only some objects are allowed to pass through.
Sure, absolutely. So we talked about that. That's semi-permeable. Some of your books might say selectively permeable. Yeah, only certain things are going to be able to readily, pass through that membrane without assistance. Okay, so we talked about some mechanisms of getting things across that membrane. We talked about diffusion, osmosis, endocytosis, exocytosis. Those types of mechanisms are going to be necessary, again, because of the fact that this is a semi-permeable barrier. Okay, yeah, so we talked about osmosis, which is basically a form of diffusion where molecules, water molecules, can easily move across that membrane. There are other molecules that won't be able to move across so easily, and they will have to be facilitated by like carrier-mediated transport and so forth. And that's where those membrane-bound proteins come into place. We talked about some of the jobs that they might have in terms of functioning as ways to get things across the cell, right? Functioning as channels where larger molecules can be transported across, such as glucose. All right, so the prokaryotic cells, let's talk about this cell wall for just a second. So I mentioned the cell wall earlier. It's this outer barrier right outside of the plasma membrane that we are going to find in bacteria cells, and it's mostly made up of peptidoglycan. Okay, write down that word peptidoglycan. All right, this peptidoglycan becomes very important when we're trying to identify bacteria. Okay, so if we're in the lab and we're trying to examine a bacterial culture and we're not really sure what it is and what organisms might be in this mixture, we'll do what we call a gram stain. Okay, so a gram stain is a very simple procedure where we can basically carry out it's like a staining. We use some dyes and then we visualize that microscope slide under the microscope. And depending on what color those slides appear, we can identify categories of bacteria, and we can identify them as either gram negative or gram positive. And so this peptidoglycan layer gives us some information because the dye or the stains that we use for this process is going to respond differently depending on whether or not we have a thick peptidoglycan layer or a thin peptidoglycan layer in that cell wall. Okay, so we're saying that all bacteria are going to have this cell wall structure present, but in some bacteria, it can be a very thin sheet, or in some others, it can be a very thick cell wall. Okay, and if it's a thick cell wall, when we carry out that gram stain procedure, the crystal violet primary stain is going to be retained after we go through our series of washes and so forth. Then when we look at that slide under the microscope, we're going to see sort of this purple violet color cells. Okay, so that's going to tell us a lot about our bacteria population. All right, if it's purple. In contrast, if this is a gram negative organism, it's going to have a very thin peptidoglycan layer. And so when we go through with those same steps for the gram stain procedure, and we go through our washes and so forth in that counter stain, the cells are going to wash off that crystal violet color, and they're going to appear sort of a pinkish red color under the microscope. And so we'll then determine that those are going to be gram negative organisms being present. And as you, your next class, when you matriculate into microbiology, if you take that next semester, we'll talk a little bit more about, how to look under the microscope and identify what we think we see based on not only the color of these cells, but also the morphology to try and further delineate what organisms might be present in this culture. So this is a picture that's stained of E. coli. Okay, I can look under the microscope. I can look at the morphology or the shapes of these cells that are here. I can see that they stained sort of a pink red color. That lets me know that crystal violet washed off and it retained the counter stain, saffronin. And this is likely going to be a culture that contains gram-negative bacteria such as E. coli. Okay, while this one is gram-positive because it's nice and dark. It retained that crystal violet, the primary stain, during the gram stain procedure. So again, knowing, carrying out a gram stain procedure can tell us a lot of things depending on what our objective is. But the cell wall is the limiting factor here in being able to like quickly and readily identify unknown bacteria in a culture. This is just a little bit more detail about the peptidoglycan that's found in the cell wall of bacteria. You'll learn more about this on next semester. I'm not going to require you to know all the intricate details about the structure and the assembly of this peptidoglycan cell wall. I just want you to know that the structure is composed of this particular polysaccharide. All right. All right, and again, this is just another illustration of what a gram-negative organism might look like versus a gram-positive in terms of the cell wall. So this blue layer right here represents the cell wall. All right, this would be our plasma membrane in green. All right, but the structure immediately outside of it is our cell wall. And in gram-negative bacteria, on your notes, you should be indicating that gram-negative bacteria are going to be characteristic of that thin peptidoglycan cell wall structure, and gram-positive are going to have a thicker peptidoglycan layer, and that gram-positive bacteria will retain our primary stain more if we were to do a gram stain procedure. And we collectively refer to the barrier that's created with that plasma membrane and the cell wall as the cell envelope. Okay, so that cell envelope is very integral in protecting that bacteria cell. I think that's kind of all I wanted you to know for prokaryotes. Yeah, you'll get a lot more of this next semester with us in microbiology. I'm gonna stop the share of this one. I want to turn our attention over to viruses for a minute. What questions do we have about prokaryotes before we go over to viruses? Any questions? No questions? Awesome. All right. I'm going to share my screen again. Let's take a look at viruses. All right. Now, viruses, again, you've already read and kind of annotated, so some of these things should stick out to you right now. Viruses are very unique organisms, if we can even call them organisms. Some books would probably refer to them as infectious particles, if you will. But these organisms are unique because they are obligate intracellular parasites. They are certainly parasites for the simple fact that they require a host. Okay, so by definition, we'll refer to them as an obligate intracellular parasite because they can only survive intracellular environments inside of a host cell. Okay, obligate intracellular parasites, meaning that they can only survive for extended periods of time inside of a host cell. And that host cell can be any organism like bacteria, animals, plants. There aren't very many organisms that are going to be resistant to a virus coming in and setting up shop. Okay, so we're going to look at some various viruses today: the bacteriophages, animal viruses, plant viruses. When we're looking at the virus in comparisons to other microorganisms, these are very, very small, tiny organisms. Okay. And again, there's debate about whether or not these are even living things, because outside of a host cell, okay, they're inert. They don't have any enzymatic metabolic activity that is required to carry out life processes. Okay. In our first unit in this course, we talked about characteristics that are required for life, and that every single organism that we consider a living thing has to be able to carry out these processes, utilizing energy, having metabolic activity. Viruses can't do that on their own. Okay. They have to be inside of a host cell. And so this is just a visual to sort of give you a point of comparison with the size of a virus. This is the size of a typical eukaryotic cell. It looks pretty big, right? And we know we have the nucleus on the inside and so forth. And then we have a point of comparison right here with prokaryotic cells. We can still see that there's a significant size difference between this prokaryotic cell and then this black dot here that represents a typical virus. Okay, they're extremely small in size, so much so that if we were trying to visualize a virus in the lab, we have to use a special type of microscope. We can't use the typical compound light microscope that we talked about for visualizing the things that we would do like in a regular teaching lab or microbiology lab. We use a compound light microscope for magnification. That won't be sufficient for looking at viruses. We need something with a higher resolution, and you write this down. We use a electron microscope. All right, we need the electron microscope to properly visualize viruses in the lab. Okay, so the compound light microscope would not be sufficient because these are extremely small organisms. All right. So just in terms of terminology here, this term virion refers to a complete viral particle. Okay, so one virus or viral particle is called a virion. Write that word down, virion. Okay, by definition, a virion is an intact and infective virus particle. Okay, and so in order to meet those requirements, we got to have two things present, two critical components here for an intact virion. All right, we've got to have that nucleic acid, right? And so viruses, their genetic material can be either DNA or RNA. Okay, so we have the nucleic acid, and then we have this protein coat or this capsid that's going to be formed around the nucleic acid, and sort of that's going to protect it. Okay, this protein coat or capsid, all right, that encloses the genetic material of the virus. Okay, so those two components are required for us to have an intact virion. And then some other viruses can have additional structures such as the envelope, spiked proteins. It just depends on the virus. Like we see here in the influenza, we're going to have those spikes present that can aid in attachment and other functions. Okay. So yeah, so an intact, so a single viral particle is known as a virion. That's one infectious viral particle. These are just some images and micrographs of various viruses that we know about. This tobacco mosaic virus, this is a plant virus. Adenovirus, that's an animal virus. Influenza, you're definitely familiar with it, and you can see these spikes coming off of these off of this flu virus. These are glycoproteins. This is a bacteriophage, and we'll talk a little bit more about that bacteriophage in just a moment. But this should be, we'll spend some more time with this in microbiology. But just from if you saw these structures around, like, you should be able to readily identify this structure as a bacteriophage, where you have that head region and a tail component. I think we'll talk about that a little bit more in just a second. But again, in terms of viruses being infectious particles, we got to have that nucleic acid, and that nucleic acid is going to be surrounded by this protein capsid. Okay. Viruses, just like bacteria, okay, those prokaryotes that we just talked about, they have different shapes or morphology. Write down the term morphology, okay? We can identify types of viruses based on their morphology, how they are made, what does their shape and structure look like. That is the same for bacteria. All right. When you take microbiology, we'll definitely spend some time looking at various shapes and morphology of bacteria. So again, if you're looking under the microscope, you don't really know what's in this culture, but you can identify some various shapes. You can look at the gram stain procedure and so forth, and you could pretty much have an idea of what organism that might be on that microscope slide. Okay. So likewise, we can use electron microscopy to also identify viruses based on their structures. Okay, some viruses can have the helical morphology. Okay, so the TMV, tobacco mosaic virus, or Ebola. You heard about Ebola before. Some have a polyhedral arrangement. Some might have an envelope. Others can be more complex, such as the bacteriophages that are going to have that head and tail and that sheath area. And we'll look at it a little bit more in a moment. Here is a bacteria where there's a head and a tail. All right. And so again, I'm not expecting you to get all of the intricate details right now about viral morphology, but I want you to understand what it is and the fact that we can identify viruses based on their morphology and arrangement. And so here are some common viruses that, not common, Ebola has resurfaced, and we never even imagined that we would have to deal with something like Ebola in our lifetime. But Ebola is a virus that we have to deal with the helical arrangement. Polio virus, this is one of the ones we've been able to sort of not necessarily eradicate, but yeah, we've been able to keep it at bay due to, I think we can say eradicated. I think we start to see more polio-like viruses right now, but I think polio itself has been eradicated due to vaccination. And so what we're hoping for right now with this coronavirus pandemic that we're in, we're hoping that we will soon have a vaccine that can help us to control the spread of the virus. Here's another one, influenza. This is the virus that causes the common flu. All right, this is an enveloped virus that has these glycoprotein spikes on the outside. Bacteriophages. All right, these are unique viruses. Bacteriophages, as the name suggests, are viruses that specifically infect bacteria. Write that down. If I asked you about a bacteriophage, if nothing else, you should be able to tell me that bacteriophages, and some people will refer to them as phages. Phages are viruses that only infect bacteria. Okay, they have a very distinct morphology. Okay, they've got this head region, this sheath kind of neck region, and then there's this tail region. Okay, this is what a phage looks like under the microscope. This is an electron micrograph of a bacteriophage. Yeah, we'll talk about bacteriophage in a little bit more detail in just a second. But just like we cultivate and study bacteria in the lab, we do the same thing with viruses, okay? But in order to cultivate viruses in the lab, we need living cells, right? We said that viruses cannot carry out, the properties that are required for life by themselves, okay? So if we contrast that with bacteria cells, you've seen petri dishes in the lab. You've probably seen a picture of a petri dish before. Well, we use those, and we can put basically artificial nutrients in those petri dishes, and we'll call it a plate. We call it plates with microbial media on the inside. And it's basically these microbial media are like nutrients or food that these bacteria cells need to grow on, and we can grow those bacteria cells directly on those petri dishes. Well, we can't do it that way entirely with viruses. We've got to have some living cells, okay, to serve as the host in order to be able to cultivate and grow this virus and study it in the lab. Okay, so we need things like tissue culture. okay, or eggs, embryonated eggs, or bacteria cultures in order to grow viruses. And that's what this word cultivate means. It means to be able to grow it in the lab for, research purposes, to be able to study various characteristics and so forth. So we cultivate them in the lab, and it's basically sort of creating this optimal environment that we know they'll grow in. Okay, so again, we're talking about the cultivation of viruses. This is a snapshot of what a petri dish looks like. But on this petri dish is bacteria growing. Okay, and so in terms of bacteriophages, we're cultivating bacteriophages by spreading bacteria out on this plate. Okay, and so all that cloudy area in this petri dish is a lawn of bacteria. And then we see like these clearing zones, like where there are no bacteria growing. And that is because that's where we have phage present. Okay, bacteriophages basically kill once they infect the bacteria cell. They can replicate to the point of causing that bacteria cell to lyse or burst open. Okay, and that becomes evident when we start to see these clearing zones in our petri dish. Okay, that means viruses are present in these areas because they have caused those bacteria cells that would be present right there to burst open or lyse. Okay, so this is an example of trying to cultivate viruses. In this case, we've grown some bacteria on this petri dish and inoculated it with virus. Okay, and the evidence that these viruses are present are these clear zones, okay, on our plate that we call plaques. So plaques let me know that there's some bacteriophage somewhere on this plate, and they're particularly where these clearing zones are. All right. Viral replication, like bacteriophages can take on two different life cycles once they have infected their host. Okay, they can take on a lysogenic phase. Write this down. I do want you to know these two things. Bacteriophages can undergo a lysogenic pathway or latent cycle, where they basically infect the host and they just kind of lie dormant for a long time. They don't cause any problems right away. Okay, this lysogenic cycle results in, again, the bacteria genome integrating with the host genome, and it's just going to exist there for a while. However, during that lytic phase or this lytic pathway, this one is going to cause some destruction pretty quickly. Okay, these types of viruses get into the host cell. They're in these bacteria cells and they're replicating quickly. And they're replicating so much so that they're going to eventually cause that cell to burst open. Okay, that's what's being represented right here. The lytic pathway is going to result in host cell lysis. It's going to burst open and more viral particles will be released. Okay, so I do want you to be able to briefly sort of describe the lytic and lysogenic phase of a bacteria phage. Okay. And so the animal virus life cycle, again, there's some other things that have to happen in order for an animal virus to be able to get in and do what it needs to do. It's got to be able to penetrate that cell membrane, right? So attachment, penetration, uncoating, biosynthesis to synthesize the viral specific proteins and so forth. There's a number of things that need to happen. I'm not going to challenge you with knowing all of the intricate details of that, but understanding that, again, various, depending on the virus, there's going to be different steps in that life cycle that has to happen in order for that virus to be successful in penetrating the host. All right, I think we'll stop right here. I want you to be able to not get overwhelmed by this information, but I want you to know some of these basic characteristics and points of comparison between, like a prokaryotic organism and a virus and eukaryotic organisms, okay? Certainly the most important characteristic here in terms of the virus is that it's an obligate intracellular parasite, which means that it just cannot survive outside of a host. Okay, so in order to demonstrate or carry out any of the characteristics that are required for life, viruses must be inside of a host. Okay, that host can range from any different organism, such as a plant, an animal, or bacteria. You should certainly have a great point of reference in terms of size, in comparison between the size of a eukaryotic cell, the size of a prokaryotic cell, and the size of a virus. All right, I gave you some terms for you to be familiar with. What is a virion? What is a bacteriophage? Okay, what does the term morphology mean? Okay, morphology refers to the arrangement or the shape or structure of the virus. Okay, we can certainly identify viruses based on their morphology. Okay, I gave you, some examples of some of the viruses that we've studied or dealt with in history. I talked a little bit about cultivating viruses. The main point I want you to know as far as cultivating or growing viruses in order to study them is the fact that we have to have a living tissue. We have to have living cells in order to grow these viruses. We can't just grow them on microbiological media like we do bacteria cells. Okay, and then this was an example of studying bacteriophage activity, okay, by growing the bacteria in this petri dish and then inoculating the virus and checking for the presence of these plaques here, which are these clear areas that let me know that viral particles are present on this petri dish, okay? So, and then also the bacteriophage life cycles, okay? I want you to be able to talk about this lytic and lysogenic pathway, like what's happening. Okay, one is where it sort of integrates into the host genome and it can kind of lie there until any further disturbance, and then it can flip over to the lytic pathway where it's basically going to result in host cell lysis. Okay, all right, I'm going to stop right here today. You have your concept review questions. What I want you to do this evening is to go back now to